DNA, deoxyribonucleic acid, is a polymeric molecule that carries genetic instructions for the development, functioning, growth, and reproduction of all known organisms in many viruses. You can think of it as a recipe to build an organism, step-by-step instructions for every biological process. DNA is a long chain of building blocks called nucleotides: adenosine, thymine, cytosine, and guanine. Similar to how information is stored in ones and zeros on a computer, information is stored as a combination of nucleotides in DNA.
The instructions contained in the DNA are executed in every cell over two major processes, called transcription and translation. A theory called the central dogma of molecular biology describes how these work. In transcription, the information in the DNA of every cell is copied into small, portable RNA messages. Short stretches of DNA are copied into RNA as and when necessary. For example, a plant may need only certain genes during the flowering season. RNA copies or transcripts of these genes are only created during that season.
RNA, or ribonucleic acid, is a polymer similar to DNA but much less stable. Its nucleotides are represented as A, U, C, and G. Because of its relative instability, it is mostly used to communicate information from DNA to various parts of the cell. However, many viruses, including the SARS-CoV, use RNA instead of DNA to store information. Translation is the production of proteins from RNA transcripts. These RNA molecules are recruited by cellular protein factories, ribosomes, which then piece together a protein according to the RNA sequence. A combination of 3 nucleotide bases goes for one amino acid. Many amino acids are put together to form proteins. These proteins are the source of all functions. They can act as enzymes or hormones or structural support depending on their structure. Producing the right protein at the right time is the key to stable life.
There is one process where we deal directly with the precious DNA itself, DNA replication. Every time a cell divides, it needs one copy of DNA for each daughter cell. The perpetuation of life is possible only due to this copying process. The central dogma of molecular biology is the statement that these processes are the only ways in which genetic information is processed, and that this transfer of information is one way, from DNA to RNA to protein. Some viruses, however, are exceptions to the central dogma. A few use RNA as their genetic material and can replicate it directly. Others, called retroviruses, actually make DNA from RNA.
Regulation of gene expression involves a wide range of mechanisms that are used by cells to increase or decrease the production of specific gene products by controlling the rate or quantity of the products in the above steps. There could be many things that could influence the rate of gene expression, including light, pH, temperature, presence of certain chemicals such as IPTG or alcohol.
Synthetic biologists take advantage of these mechanisms to slow or speed up production of items they require. For example, when they use genetically modified organisms to produce a chemical on an industrial scale, they could try to see which of these factors should be provided in the right amount to speed up all the processes in the central dogma and get the maximum yield.